Life on Earth exists only because the sun provides energy in the form of light. Living organisms trap, store, and use energy to maintain healthy cells. Plants and most other photosynthesizing organisms have chloroplasts, specialized cells which trap the sun's energy and convert it into chemical energy.
This is a process called photosynthesis, the process by which plants, algae, and some bacteria use the sun's energy to chemically convert carbon into carbohydrates such as sugars and starches. Byproducts of photosynthesis include oxygen, molecules of ATP, and heat. Some of the energy-rich compounds created for photosynthesis are used immediately, and some are stored for future use.
But how exactly does photosynthesis work? In these processes of photosynthesis, water serves as a source of electrons, and the oxygen byproduct is released into the environment. The light-dependent reaction occurs on the cycloid membrane of the chloroplast. To start the whole process, light strikes a pigment molecule in the photosystem 1. Energy is then transferred to the reaction center of photosystem 2. The energy is used in photosynthesis, which splits the water molecule into protons, which are released into the environment, and electrons, all of which are transferred to photosystem 1. Electron carriers transfer the electrons to photosystem 1, then more light strikes pigment molecules in photosystem 1. Energy from is then transferred to the reaction center of photosystem 1. A series of electron carriers transfer electrons from the reaction center's molecules to NADP, allowing NADP to combine with hydroxide ions to form NADPH. NADPH is stored in the stroma, ready to be used in the Calvin cycle.
Chemiosis occurs across the cycloid membrane of the chloroplast. Its purpose is to move protons from a region of high concentration on the lumen side to a region of low concentration on the stoma side. The energy release is used by the enzyme ATP synthase to form ATP.
The Calvin cycle is the next and last step for photosynthesis. The Calvin cycle will begin with 3-ribulose biphosphate.
The first step is carbon fixation. An enzyme called Rubisco will attach carbon dioxide to each RUBP and forms 6-phosphoglyceric acid and 6-PGA.
6-PGA will immediately split into 3-PGA, each with a total of 6-3-PGA.
The second step is carbon reduction. 6-ATP and 6-NADPH give energy to generate 6-GP3.
This process is called reduction because ATP will convert it into ADP, losing phosphate atom to G3P, same as NADPH will convert into NADP, losing both energy and hydrogen.
Third step is carbon regeneration. 1 G3P will be released from the cycle, while the 5 remaining G3Ps that has a total of 15 carbon molecules will form another 5 carbon molecule called RUBP to begin another Calvin cycle.